The chemical compound ammonia is an important source of nitrogen in the human body, but can be toxic if found in high enough concentrations in brain tissues, causing hyperammonemia, a condition that can lead to brain damage and death. Amino acid catabolism resulting from the ingestion and breakdown of proteins results in great amounts of waste ammonia, which could build up and reach toxic levels in the body. Because of this, the body needs a way to both detoxify surplus ammonia and excrete it. Thankfully, the toxicity of ammonia does not generally cause problems for humans due to the pathways in the body that prevent it from building up. The enzyme carbamoyl phosphate synthetase, or CPS, expressed in the mitochondrial matrix of small intestine and liver cells, can convert ammonia to carbamoyl phosphate using substrate channeling. CPS synthesizes carbamoyl phosphate from 2 molecules of ATP, 1 molecule of bicarbonate, and 1 molecule of glutamine or ammonia. The carbamoyl phosphate produced is subsequently used in 2 biosynthetic pathways: the synthesis of arginine or urea if it reacts with ornithine, or the synthesis of pyrimidine nucleotides, namely cytosine, uracil, and thymine, if it reacts with aspartate. This makes CPS an important enzyme in the urea cycle, used in most terrestrial vertebrates to remove waste ammonia from the body via excretion. The reaction catalyzed by CPS is as follows. ATP is first used to add a phosphate group to bicarbonate, making carboxyphosphate and releasing an ADP molecule. Carboxyphosphate then reacts with ammonia, likely released from glutamine, an amino acid used to transport ammonia in the bloodstream without it being toxic, to form carbonate, releasing inorganic phosphate. Finally, the second ATP molecule phosphorylates carbamate, forming carbamoyl phosphate. The interest in investigating CPS is due not only to the reaction it catalyzes, but also its unique characteristics. CPS is an approximately 159 kilodalton heterodimmer consisting of 2 non-identical subunits, the 41 kilodalton glutaminase subunit, which has 1 active site, and the 118 kilodalton synthetase subunit, which hosts 2 other active sites. The 3 active sites on the enzyme are spaced surprisingly far apart, requiring the usage of a molecular tunnel to connect them, which measures roughly 100 angstroms in length, and is noted here in blue. The active site of the glutaminase subunit contains a glutamine binding site and catalyzes the hydrolysis of glutamine to glutamate and ammonia. The synthetase subunit has 2 carboxyphosphate domains with ATP binding sites. The first of these sites, located at the N-terminus of the subunit, catalyzes the phosphorylation of bicarbonate, while the second site, located at the C-terminus, catalyzes the phosphorylation of carbamate. Since both ammonia and carbamate are unstable reaction intermediates, they can be difficult to transport in the cell, and so their movement needs to be regulated. To facilitate the movement of such substrates, substrate channeling, the passing of an intermediary product of metabolism from one enzyme to another active site without releasing it into the cell, is employed. Substrate channeling has been observed in proteins, CPS included, through X-ray crystallographic analyses. Substrate channeling has many advantages, including the protection of unstable intermediates or products, the sequestering of toxic molecules, and improved catalytic efficiency of the enzyme. Without carbamoyl phosphate synthetase and the vital role it plays in amino acid and nucleotide synthesis, as well as the urea cycle, protein and nitrogen metabolism as we know it would not be possible, and hyperammonemia would be much more common. Similarly, our current understanding of carbamoyl phosphate synthetase's function and structure would be impossible without the scientists who have dedicated their time and energy into studying it over the past 60 years. I would like to thank those scientists and acknowledge those whose work I referred to for the information necessary to make this video. As well as being displayed on the screen currently, links to the specific papers cited are in the description below.